So last week we did about the nutrition of microbes, no? So today we'll do about the growth of microbes or the growth of bacteria. Okay, so to grow they require proper nutrition. How they obtain their mode of nutrition, we saw, no? Some are chemosynthetic, some are photosynthetic, right? Okay, so they need nutrition, okay? They will need elements and organic growth factors, okay? Elements means phosphate and the inorganic one, the air, the sunlight, the water, okay? These the inorganic things. These are the minerals, okay? Organic growth factors are the dead debris of plants and animals, okay? I'm organic, you are organic, okay? We will decay one day, no? So the first type is the essential elements. They are the nucleic acid, protein, fat, carbohydrate. So the main structural, structural compound of any living thing, like you and me, I have DNA, you have DNA. So DNA is made up of nucleic acids, no? Then from the nucleus, from the DNA, we will synthesize the proteins, okay? Proteins are, example is like the enzymes, no? Enzymes in my body, okay, which will digest the food, okay? So the proteins, then the carbon, the elements that build up my body, no? Like my nails, my skin, my hair, it's fully made of carbon, right? Similarly with plants and animals, okay, we are all organic compounds, no? Organic means those which have carbon in their structure, okay? Either the alkenes or alkynes, we are all having this carbon structure. Okay, so the essential elements that they require will be carbon, nitrogen, hydrogen, oxygen, etc. Okay, because these are the building blocks of your nucleic acid. Okay, DNA. No, remember the structure of DNA. Then you have the phosphate group. Then you have the nucleic acids. No, that combine with each other. Okay. Then there will be other elements like sulfur, phosphorus, etc. So the minerals, the calcium, magnesium, potassium, iron, etc. These are required in trace elements. Okay, so like you and me, I need also iron in trace element. Why? Because in my blood, the heme, no, my blood, it's connected with the iron compound. No, okay, from the iron, it binds the iron in my blood. It binds to the oxygen. Okay, so. So those who are photosynthesizing, they will need copper or manganese, right? And they may need zinc and all the trace elements. So organic growth factors, they will get vitamins, okay? They will get vitamins from the dead and decaying matter of the living things, okay? Okay, let's look at the environmental factors. The water, the temperature, the pH. Okay, all here. Okay, you go through by yourself since the note I've already sent. This is just the easy, like school level, no? Water you require. 80% of cell volume is made of water. Okay, like human, I am 70% water. You also, you're 70% water. Then oxygen, depending on whether they are aerobic or they are anaerobic. Okay. Then carbon dioxide, those which are photosynthetic, they may require. The temperature, depending on where they live, whether they like very high temperature, those that live in volcano, they will be thermophile. Those who are growing like us, you know, in the middle, not so hot, not too cold, they're thermophile. Then very low temperature will be psychrophile. Okay. Then the pH, it depends, okay? It depends. Those who cause disease, they prefer human pH, no? 7, around like 7. Okay, there are some that require 10, etc. You can look by yourself. Then light also, some they need, some they don't need. Then osmotic pressure, depending on where they are found and what they require. Okay, so let's go to the important type, the important part of this lecture. So, okay. So we saw the binary fission, no, in the last 2 classes back, where from 2 you get 4, etc. So there is a term known as generation time. It is the interval of time between 2 cell divisions. Okay, that is your generation time. It varies from species to species. Then bacterial count is the total count. It's the total number of bacteria in the medium, which include both the living and the dead cells. Okay, so this is your like net question, no, multiple choice question. What is bacterial count? It is the total number of bacteria in the medium. Okay, dead and alive. Okay, you'll get option no, living. Bacterial count is the number of living bacteria A. Then number B, number of dead bacteria. Okay, then C will be only living without the dead. Then D will be both living and dead. So those are the type of question that can come. Okay, let's look at the more important part is the bacterial growth cycle. Okay, so okay, let me just give you a small introduction. Suppose I want to grow bacteria. Okay, then when you want to grow a child, you have to give it proper milk, no, proper food, nutrition, right? Then proper space to grow in. No, it needs a room, it needs a cot, right? It needs food, it needs resources. Okay, so similarly with bacteria, if you want to grow it in lab, you have to prepare in sterile condition, means in laboratory condition, no, where you have sterilized the utensils that you're using. No, you have sterilized the utensils with alcohol. No, now that it's the normal, no, using alcohol to sterilize our hands and all, exactly like that. Okay, so you prepare the food. Let's say this is a petri plate and you filled it with the food. Okay, let's say in this conical flask you added water, then you added the sugar, okay, the glucose, then you added the vitamins, you added the minerals. Okay, then you want to grow it in a semi-solid medium, you want to add then you can add agar. Okay, then this one you pour into this petri plate. Okay, so you poured it around this much, then you waited for some time, it became solid. Then with a let's say a knife, not a knife, like a brush, a sterile brush or forceps. Okay, you just streak here, you just touch it here, and then you close the lid. Okay, let me draw clearly. Then you close the lid of the petri plate and you wait for 1 month or maybe 2 weeks. Okay, after 2 weeks you will see white or brown. Okay, so you have noticed that your bacteria, they have grown. Okay, or your fungus, they grew. Okay, so you have provided the food, then you provided the space for them, the petri plate, no, you provided the temperature, you provided everything. Then you left it after 2 weeks, you left it again another 2 weeks. Again the culture grew from that size. Okay, it grew this much, almost full. Okay, then what did you do? You left it again another 2 weeks, another 3 weeks. It's already full, no? So in maybe let's say 2 weeks, it was already full. There was no more space. Okay, no more space for the bacteria to grow. It's already full. Then what happens? You will see that suppose this bacteria is a white, it has a white color like a cotton looking. It will start to die off. You will notice some parts are turning black. Okay, they turn black and they die off. Okay, let's say this part is dying off. They die off. Why? Because the food is finished, the shelter. Can I throw you all the 70 of you, can I lock you up in one small room? You know, it's already so congested. You will run out of oxygen, you will run out of food, you will run out like that. No, there are too many of you. So same here. These bacteria, they'll start dying off until maybe just a few are left. Okay, maybe just a few are left. They have died. All this have died, and maybe just 1 or 2 is left. Okay, so the same way, if we humans, we are kept in one small room, okay, all 70 of you are kept in a small room, and then I said, here is 50, 50 jugs of water and 100 packets of food, okay, you survive in this in 2 days. You can survive 2 days. Then I say you survive in the same amount for another 3 days. Then you will start to reduce your intake of food. You will not eat so much. Then I'll say you have to continue till surviving till maybe 7 days. Then the food has finished. Okay, food all finished. Then some will start getting sick and dying off. No, same like that. Okay, so let us look at the bacterial growth cycle. So when a sterile liquid nutrient medium is inoculated by a particular bacterium, the bacteria will undergo multiplication, obviously, and it will increase in number. Okay, by that binary fission, no, it will increase in number. Okay, then let us plot a graph and follow how they grow. Okay, you understand? You already know what is happening. You already know that they are going to increase in number. They're going to multiply, right? So let us follow them from the in a graph. Okay, there are 4 main stages in a growth curve. Okay, I don't know. Oh, there. See, so here at this stage, let's say this is the number here. In the y-axis is the number of bacteria. In the x-axis is the time in hours. Okay, let's say this is 1 hour, 2 hour, 3 hour, 4 hour, 5 hour. Okay, and number of bacteria. Let's say we started from. It should be even, no? From 2 to 4, 4 to 8, 8 to what is it? 8 to 30, 16, I think, no? 2, 4, 8, 16. My maths, I have forgotten. Okay, I think it's correct. Okay, so now let us see. So, first we started only with 2 bacteria. Okay, there. Let me just delete. Here, we started only with 2 bacteria. Then it grew, no? Then now the number of bacteria will be 4 after, hypothetically, 1 hour. Then, hypothetically, after another, not 1 hour, half an hour, 1.5 hour, 1.5 hour, we got how many? 8 bacteria. Then after 2 hours we got 16 bacteria. Then at 16 bacteria you are going to see a stationary. Okay, means here when there was just 2, 4, 8 and 16, it could grow quickly. Okay, there was more than enough food, more than enough space, more than enough shelter for everyone. Okay, so they increase in number very quickly. Then what happened when they reached 16? They thought, oh God, if we 16 multiply, we will be directly 32. So directly from 16 to 32 will be too much. Okay, it will be too much. Our house, it cannot support. The Petri plate cannot support. The shelter cannot support. The water will not be enough. The food will not be enough. The nutrients will not be enough. So they decide to stabilize themselves. Okay, stabilize themselves means they try to remain at 16. Okay, means okay, there were 16, right? Okay, some will multiply, no, some will multiply. Let's say from 16 they multiply to 20. Okay, then if you minus 16 and 20, how much is it? You minus 4, means 4 old bacteria will die off. Okay, so throughout 3 hours, 2.5 hour, 3 hour, 3.5 hour, they try to maintain at that same population. Okay, 16, they reach 20, then 4 old bacteria died. Then another bacteria, they produce 4 more children, then another, produce 8 more children, then another 4 will die. Okay, so dying, living, the young are growing and living, and the old are dying. Okay, so your population somehow remains stable. Okay, but you think, fine, they are being smart. Yes, they are doing family planning. Isn't it funny? Bacteria, they are doing family planning. ठीक है, they did family planning. Okay, they have done the family planning. Then you must be thinking, if they did family planning, then shouldn't they be able to survive? Shouldn't they live? Shouldn't they be living? Shouldn't they be succeeding? Why am I seeing a down? Okay, look, this part. Why am I seeing a drop? Well, yes, they have done the family planning. Your thoughts are fine, but you are forgetting that the space and the nutrition did not change. It is not that those who died, they provided nutrition, they provided water, nothing like that. Okay, so when they died, they produced toxic. Okay, I forgot to mention that as a bacteria, they are growing, living inside that plate. Aren't they producing some toxins? Like you and me, we produce urine, we produce poo-poo. No, we poop, we urinate, okay, we vomit, we produce mucus, okay. These are our excretory products, which will be toxic if we consume them again. If you consume urine again, you will concentrate your blood with nitrogen, okay, with ammonia. It will kill us, make us toxic. So same here, I forgot to mention that not only did they grow and they took away all the food, took away all the space, but also they produced they excreted some toxic substances, their excretory products or their death products, so the accumulation. Okay, so looking at this graph again, you will notice it still went down even though the population was maintaining its same number. The thing is, neither did a new space introduced. Okay, there was no new space introduced. Neither was there new shelter, new food, and there was a lot of toxic substance accumulation. Then those toxic substances, they tend to kill off. Yes, you might have control your population, but the toxic substance which was produced by the oldest bacteria were already there. They keep on producing toxic substances as they die off. So that toxic substance, okay, toxic excretory products, they will kill even the young ones. So you will see that their population will come down. So here, 1, 2, 3, 4 hour. No, so here in 4 hour, okay, you are seeing a decline. We were at 16. No, then in 4.5 hour, we were at 8. Okay, then in 5 hour, we were at 4. Okay, then you will see. Here. Okay, here. Then you will see here, it will sort of stabilize again. Then when there were only 2 left, somehow it stabilized again, okay, because just 2, somehow they can survive, okay. All right, just like the pandemic, you saw, right, some people, they died, but some, they are so resilient, they survive, okay. Some very old woman, 85-year-old, she still could beat the virus, okay. So this, it applies to all of life, okay. Some of us will be vulnerable to this virus, yes, but still the population will never fully die off, because there are some who will be resistant, who will be resilient, okay. That fighting spirit, the fighting spirit is so strong, okay. So even here, the fighting spirit of these bacteria is so strong that still, even though the toxic substance still accumulated, they still maintain their population at that much. Okay, so I think this graph is clear. So let me just read out. Okay, so first phase, okay, first phase, this one, when we just started, okay, when we just started, is called as the lag phase, okay, LAG, the lag phase. So let us go. Okay, so the lag phase is after you inoculated or after you added the new bacteria into the sterile nutrient medium. Okay, new person into a new place. You got it? Okay. Then, so you girls, you came from different schools, right? 11, 12, you came from different schools. Then suddenly it is time for you to move on to a college. Okay, then some of you came to our college, and then some of you still behave as school children. You still bring pencil box, then school bag, then you bring lined copies, no, and then what to say, color pencil, you're still bringing. Okay, still being like a school girl. You're still wearing like a school girl. You're still tying two ponytail. Okay, obviously I know you girls are stylish, but hypothetically, okay, this is just a theory. Then you start looking around because it's a new place. No, you don't like it. I don't feel comfortable this college. It's so lonely. Okay, then after a week you made a friend. After 2 weeks, that friend introduced you to more friends. No? Then after a month, oh God, you girls are so noisy. My head is about to explode. You're laughing. You're enjoying. You're joining in the college week. You're making dramas, making plays, and making costumes. No, fully enjoy it. Means you took time to adjust to the place. You took time to adjust to the environment. So here also during the lag phase, that is when the bacteria will adjust to its environment. Okay, when it is finally settled, then it will really like grow well. Okay, just like you girls, after you settle into the college life, you enjoyed and you really were soaring high. You took part in the debates, in dances, in extempore speeches. You really expelled, not expelled, you really excelled. Okay, so here, this is when the acclimatization occurs. Okay, so at that time, enzymes and intermediate metabolites are produced. Okay, so it's when he's adjusting, okay, he will be producing all the things necessary to digest the medium. Okay, digest the food. Okay, because at first he doesn't know what kind of food he will get. Okay, when you put him in the nutrient medium, in the nutrition place, nutrition sources, then he realized, oh, there's glucose. Okay, then I'll produce cellulose. No? What enzyme we use to digest glucose? Amylase. Nee, how stupid is Miss? Yes, my brain, I don't know what happened. Then it realized I have to produce amylase. Okay, then enzymes and then other necessary metabolites. Okay, so it will reach a critical mass, means it will not reproduce as in it will not divide, but it will grow in size. Okay, grow in critical mass. It will grow in size means it's accumulating all the enzymes and metabolites that is required for it to succeed well and to digest the media properly. Okay, so in this stage there is no multiplication happening. Now the next phase, or the exponential phase, or the log phase. So this part, okay. Now the phase where you are really excelling in the college. So in this phase, the bacteria will undergo cell division and their population number will increase exponentially at a logarithmic rate. Okay, log rate is mean from 2 to 4, 4 to 8, that kind of like logarithmic rate. Okay, so when we plot a graph, it will give us an inclined, it will give us a slope. Okay, if we plot a graph, it will be like this, a slope. Okay, because they are increasing at a very quick rate. Okay, then we come to the stationary phase. Okay, this is the phase when it is all the nutrients have finished. Okay, this is when what happens if you suppose you kept failing. Okay, people, God forbid, you will never fail, because you girls, you understand you need to work hard. But suppose you had failures. Okay, you kept failing, you kept giving the you kept giving the exam, and it was not working out there. That is when you become stationary, you become bored. Okay, so it becomes neutral. So in the neutral phase, so the cell division will slow down, okay, due to the lack of the nutrients and the accumulation of toxic products. I've already explained in that story. Then the death will start at a slow rate, okay, and the new cells also will form. Okay, means death also slowly, new cells form also slowly, no more exponential. But somehow it will remain constant, the population will remain constant, as I told you, will remain at 16. Okay, so there will be toxins, etc., that will be produced during this stage. Okay, then we are coming to the decline phase. Okay, this decline phase. So in this phase, the total number of cells will remain constant, but the number of healthy cells will decrease due to exhaustion of nutrients and accumulation of toxic products. Okay, and very few cells will remain viable for a long time, even after death of most of the cells. So these resistant cells will be those that somehow could adapt. Okay, they somehow could eat from the remains of the dead cells. Okay, so this is done. I hope it's clear.